Welcome back, everybody. At Bridgewater State University, undergraduates have developed a device that's used in cancer research. They're believed to be the first undergraduates to build what are called optical tweezers.
And joining us to talk about it tonight are Brian Dolle, Professor Thaya Paramanathan from the Physics Department at Bridgewater, and Zach Ells. First of all, the Nobel Prize was given in this field this year. Explain, Professor, if you would, what optical tweezers are.
So we all know tweezers, which you use to grab tiny objects. So when we use light to grab tiny objects, when I talk about tiny, I'm talking about objects that you can't see by your naked eye, which you can see through the microscopes, like cells, DNA. So when we use those light lasers, intense light, to trap these objects, those are known as optical tweezers. And Dr. Arthur Ashkin, who developed this, won the Nobel Prize this year, and which opened the gate for us to do this research what we do right now.
So give us a sense of how this works. There are only 15 to 20 of these in the country, right? And how are they used to advance biological research? How do you use a device like this to research cancer?
Yeah, so basically we're able to capture a single particle within a flow cell, and we use two laser beams, and inside this flow cell we capture that particle, and we're able to basically get a DNA molecule, stretch it, and introduce it to a cancer cell or a cancer drug and see how it interacts with that DNA molecule.
So in this part right here, you'd have a DNA molecule.
Yep.
And you would then hit it with light, and you can get a sense by introducing drugs of what it's doing to that DNA.
So the laser, the light is used to grab the DNA molecule, isolate the DNA molecule. And then we introduce the cancer drug and see the difference in the stretching, as Zach explained earlier.
And cancer research is so important to so many families out there, desperate for drugs, desperate for cures. How are you able to do this at the undergraduate level? Because it's usually much more advanced work, right?
Yeah, so we have research scale optical tweezers, so we can collect data and do research there. And so what we do is, again, we isolate a single molecule of DNA. Zach and I actually are studying a drug called doxorubicin. It's been around since the 1970s. It's used to treat breast cancer and a number of other cancers as well. And so, again, we flow in the drug, allow that to bind to the DNA. We stretch it before the drug, after the drug, and then by stretching and comparing those DNA curves, we can understand the binding mechanism.
Professor, just from your perspective as a professor, two undergraduate students who are doing work that's this sophisticated, what do you have to say about that?
I can say that, I can claim at least, as no one else can claim this. So that's a pretty decent job they are doing, because we have fabulous students. The students we have at Bridgewater are hardworking students. Most of them work outside to survive. So the hard work is in their DNA. So stretching DNA is not hard at all.
I will ask, the cancer drug that you're studying, do you have results yet? Have you had a sense of how well it's working?
Yeah, so this summer we actually were able to obtain a good data sets on several different concentrations of that drug, and we actually plan to study that drug again at some other several concentrations to get a better sense of how it's binding to the DNA.
What's next for you? Graduate school, medical school?
So I'm applying right now to graduate programs in aerospace engineering, actually. I want to design and build satellites.
Very cool. How about you?
And funny that you say, I'm actually interested in medical school or biomedical engineering, so I'm interested in like the biology side of it.
Interesting.
I think these guys will go far.
Definitely, definitely. There's no doubt about it.
All right. Well, Professor there from Bridgewater State University, Zach, Brian, thanks for coming in tonight. We appreciate it.
Thank you for having us. We are delighted to be here. Thank you.
Thanks, and good luck, guys. Appreciate it. Let's